So now I will be discussing the three levels of protein structure only because the next reporter will be the one who will discuss the fourth structure. So to describe and understand the complexity in protein structure, so it involves considering the four levels of protein structure. And every protein has its own unique amino acid sequence, and that is the primary protein structure that I will be discussing. So it is the order in which amino acids are linked together in a protein. It involves more than just the numbers and kinds of amino acids present. It also involves the order of attachment of the amino acids to each other through peptide bonds. So, the British biochemist Friedrich Sanger determined the primary structure of the protein hormone insulin in 1953. And as we all know that insulin is a hormone that regulates blood glucose levels, was the first protein for which primary structure was determined, and there is 51 amino acids during the discovery. His work is a landmark in biochemistry because it showed for the first time that a protein has a precisely defined amino acid sequence. Today, primary structures are known for many thousands of proteins, and the sequencing procedures involve automated methods that require relatively short period of time. And for the myoglobin, it's actually the one of the primary structures, and as we all know, it is a protein involved in oxygen storage in muscles, and it contains 153 amino acids assembled in the particular definite order. So the primary structure DI of a specific protein is always the same regardless of where the protein is found within an organism. It's either gikan na siya sa human or gikan siya sa animal. Like for example, the primary structure of insulin in cows, pigs, sheep, and horses are very similar both to each other and to human insulin. And currently, in insulin, it cannot be taken orally because they, like other proteins, lose their activity when they encounter the stomach's digestive enzymes. There is actually an analogy that is often drawn between the primary structure of proteins and words. So, as we all know, words conveys information and are formed when the 26 letters of the English alphabet are properly sequenced. Same thing with the proteins, no? They are formed from proper sequence of the 20 standard amino acids. And if we remember the discussion sa first na nag-discuss sa protein, the introduction, it it was discussed there the non-polar to polar acidic amino acids. And just as the proper sequence of letters in a words is necessary para atong hinumdum masabtan and letters make sense also. And the proper sequence of the amino acids is necessary to make biochemical active proteins. Furthermore, the letters that forms a word are written from left to right. Same thing with the amino acids protein formulas. And I'm showing in the slides here the important points concerning peptide bond geometry are, so the peptide linkage are essentially planar. This means that for the two amino acids linked through a peptide linkage, 6 atoms lie in the same plane. So if you can see the picture here, within that plane there's actually 6 lying here. If we will count the sides, get a side, 1, 2, 3, 4, and then inside the plane is there's 2. I mean inside the that side na I2. And the alpha carbon atom and the carboxyl group from the first amino acid, and the amino group and the alpha carbon atom from the second amino acid. The planar peptide linkage structure has considerable rigidity, which means that the rotation of the group about the carbon and nitrogen bond is hindered, and the cis-trans isomerism is possible about this bond. The trans isomer orientation is preferred orientation, as shown in the preceding diagram. And the oxygen atom of the carboxyl group and the hydrogen atom of the amino group are position trans here, sa up and sa down to each other. And the net effect of the peptide bond planarity is the zigzag arrangement. For the secondary protein structure, so it is the arrangement in space adopted by the backbone portion of a protein. So there are two most common types of secondary structure. The first one is the alpha helix structure, and the second one is the beta pleated sheet structure. And the type of interaction responsible for both of these two of the secondary structure is hydrogen bonding. So it's actually between the carbonyl oxygen atom of a peptide linkage and the hydrogen atom of an amino group of another peptide linkage further along the protein backbone. So the hydrogen bonding possibilities that exist between carbonyl oxygen atom and amino hydrogen atom associated with different peptide linkage and a protein backbone. The protein backbone segments involved in hydrogen bonding can be two segments from different backbones or two segments of the same backbone that has folded back upon itself. So for the alpha helix structure, so it is a protein secondary structure in which a single protein chain adopts a shape that resembles a coil spring helix with a coil configuration maintained by hydrogen bonds. The hydrogen bonds are between amino group and the carboxyl group. To further details about alpha helix secondary protein structures are: the first one is a twist of the helix forms a right-handed or clockwise spiral. Second is the hydrogen bonds between the carboxyl and the amino group entities are orientated parallel to the axis of the helix. And then a given hydrogen bond involves a carboxyl group of one amino acid and amino group of another amino acid located 4 amino acid residues further along the spiral. This is because one turn of the spiral includes 3.6 amino acid residues, and the fourth one, all of the amino acid R groups extended outward from the spiral. There is not enough room for the R groups within the spiral. The beta pleated sheet structure. It is a protein secondary structure in which two fully extended protein chain segments in the same or different molecules are held together by hydrogen bond. So showing here in this picture, there's N-terminal end, and that is where the amino group, and the C-terminal end, that is where the carboxyl group. And this U-turn structure is the most frequently encountered type of beta pleated sheet structures. And it shows here in this presentation that the B-pleated sheet structure occurs when portions of two different peptide chains are aligned parallel to each other. And term pleated sheets arises from the repeated zigzag pattern in the structure. And further feature of the B-pleated sheet secondary protein structures are: first, the hydrogen bond between carboxyl group and the amino group entities lie in the plane of the sheet, and the amino acid, the side chain R group, are found above and below the plane of the sheet and within a given backbone segments alternating between the top and the bottom position. For the unstructured segment, so it is a protein secondary structure that is neither an alpha helix nor a beta pleated sheet, and it is essential to the functioning of many proteins. It confers flexibility to proteins and thereby allowing them to interact with several different substances, and an important mechanism for rapid response to changing cellular conditions. So, an active area proteins researchers involved learning more than the biochemical function of the unstructured portion of proteins. So, the act of binding to another protein thus bring added structure to the unstructured portion of the protein, but added structure is lost when the binding interaction ceases. So showing in this picture now or is a slide, so there are two representation emphasizing the hydrogen bonds between protein chain here, and this one the B structure representing emphasizing the pleats in the location of the R group, and it's folded here, and kanisha is a structure which showing the unstructured segment. This is the alpha helix, here the alpha helix, and the B pleated sheets. Before I will proceed to the tertiary structure protein, there's actually a good analogy for the relationships among the primary, secondary, and tertiary structures of a protein. If you are familiar with the telephone cord, so the primary structure is the long, straight cord siya. And the coiling of the cord into a helical arrangement gives the secondary structure, and the supercoiling arrangement the cord adopts after the receiver's has been hung up a number of times is a tertiary structure. If makakita mo ani nga picture here, kaning straight primary ni siya, kaning dili pa siya lukot kaayo much, that is the secondary, and the very supercoiling siya, no, sa telephone cord, that is, it refers to the, or describes siya as the tertiary. And yes, for the tertiary structure protein, it is the overall three-dimensional shape of a protein that results from the interaction between amino acid side chain, the R group, that are widely separated from each other within a peptide chain. And from the name itself, tertiary, three-dimensional shape. No, in 1959, a protein tertiary structure was determined for the first time. The determination involved the myoglobin. So, kuan po siya, no? Aside sa the primary, si myoglobin is na determined po siya under the tertiary structure protein, a conjugated protein. For the interactions responsible for tertiary structure, there are four types of stabilizing interactions contribute to the tertiary structure of a protein. So the first one is the covalent disulfide bonds or disulfide bonds, and that is the strongest of the tertiary structure interaction, results from the sulfhydryl groups or two cysteine residues reacting with each other to form a covalent disulfide bond. So, atong mahinumduman, kung mo talk about covalent, that is sharing electrons. So disulfide bonds, this type of interaction is the only one of the four tertiary structure interactions that involves a covalent bond. So siya ra jud from the word niya here, the name niya here, covalent. So disulfide bond formation may involve two cysteine units in the same peptide chain, an intermolecular disulfide bond, or the two cysteine units in different chains, and intermolecular disulfide bonds. And the covalent disulfide bonds, it's actually helps maintain the protein's overall shape and stability. For the second interaction, that is the electrostatic attraction or the salt bridges, always involve the interaction between the acidic side chain, the R group, and a basic side chain, R group. And it is an acidic side chain being negatively charged and the basic side chain being positively charged. Such side chain charges occur when a carboxyl group becomes the COO negative and when an amino group becomes the positive charge group. The interaction that occurs between the two types of side chains is a positive-negative ion-to-ion attraction. The third interaction is the hydrogen bonds. It occurs between amino acids with polar R group, relatively weak and are easily disrupted by changes in pH and temperature. So the hydrogen bonds are relatively weak and are easily disrupted by changes in pH and temperature. And for the last interaction, that is the hydrophobic attraction, when two nonpolar side chains are close to each other, common between phenyl ring and alkyl side chain. So hydrophobic interactions are common between, as I mentioned, the phenyl ring and alkyl ring chains, although hydrophobic interactions are weaker than hydrogen bonds or electrostatic interaction. They are significantly forced in some proteins because there are so many of them, and their cumulative effect can be greater in a magnitude than the effect of the hydrogen bonding. To summarize the protein structure, so for the primary structure, that is the sequence of amino acids present in a protein's peptide chain or chains. For the secondary, that is the regularly repeated ordered spatial arrangement of amino acids near each other in the protein chain, which result from hydrogen bonds between carbonyl oxygen atom and amino hydrogen atoms. And there are two common structure. First one is the alpha helix and the beta pleated sheet. For the alpha, that is the hydrogen bonds between every fourth amino acids, and for the beta, that is the hydrogen bonds between two side-by-side chain or a single chain that is folded back on itself. And for the tertiary structure, that is overall three-dimensional shape that results from the attractive forces between amino acid side chains or the R groups and that are not near each other in the protein chain. And for the interaction responsible for the tertiary structure, let us remember this, the disulfide bonds, electrostatic interaction, hydrogen bonds, and the hydrophobic interaction. Thank you for listening.